Hello friends, this is the second part of the video for second chapter of Science Second for Standard 10, that is life processes in living organism part first. In this part we are going to see the cell division, which is important life process carried out in all the living organisms, and what is the use of cell division, that we'll see here, type of cell division, and the diagrams related to this cell division will be seen here. So cell division is carried out by two ways. Why by two ways? Because we have two type of cells in our body. Main type of cells in our body are somatic cells and germ cells. Somatic cells means what? All the normal cells of the body, they are somatic cells. Or stem cells, these are the original cells of our body from which our body is made or organs are made. So that somatic cells or stem cells, they undergo cell division by a particular type of cell division. And germ cells which are present or which are produced in our reproductive system, they are also dividing by means their division is there by meiosis. So like this way we have two type of cell division. Somatic cells get divided by mitosis and germ cells get divided by meiosis. So here let's see what is the use of mitosis. Mitosis is used for the growth of the body, healing of the body. The wear and tear of the body can be done by the mitosis. Means one cell is multiplying into 2 and then 2 are again multiplying into 2. Like that way there will be increase in the number of cells. That is only called cell division. Let's see what is mitosis and the steps in mitosis. Mitosis takes place in 4 stages: prophase, metaphase, anaphase and telophase. What is the peculiarity of mitosis? That is the number of chromosomes which are there in the nucleus. That is, suppose 2n number of chromosomes are there, then cell gets divided into 2 cells which also has 2n number of chromosomes. So here you can see the first step, prophase, in which the cell is there and the nuclear membrane disappears. So in prophase, condensation of basically thin thread-like chromosome starts. Due to this, they become short and thick and they start to appear along with their pair of sister chromatids. Centrioles duplicate and each centriole moves toward each pole of the cell. Nuclear membrane start to disappear and nucleolus also start to disappear. The second stage is metaphase. In metaphase, nuclear membrane completely disappears. Chromosomes now complete their condensation and become clearly visible along with their sister chromatids, and they all are arranged parallel to the equatorial plane of the cell. Special type of flexible fibers arise from the centromere. They are called as spindle fibers. They are formed between the centromere and centrioles of the cell. The next step is anaphase. In anaphase, the centromere split and thereby sister chromatids of each chromosome separate and they are pulled apart. They are pulled apart towards the pole with the help of spindle fibers. Separated sister chromatids are now called as daughter chromosomes. Chromosomes being pulled appear like bunch of bananas. In this way, each set of chromosomes reach at the 2 opposite pole of the cell. The next stage or the last stage is telophase. In telophase, you can see that there are 2 nuclei formed. The chromosomes which have reached at opposite pole of the cell now start to decondense, and due to which they again become thread-like, thin and invisible. Nuclear membrane is formed, nucleolus also appears, and the 2 daughter nuclei are formed in the cell. The nucleolus is also seen in both of the daughter nuclei. In this way, karyokinesis, that is the first stage, is completed, and next cytokinesis begins, in which this cytoplasm is going to divide. So there is the two new cells are formed, which are daughter cells. In this process, a notch is formed at the equatorial plane of the cell, and the two cells are formed. In between, the organelles of the cytoplasm, they get replicated, and complete two set of the cells are formed. They are called daughter cells. So these are the 4 stages of mitosis. Let's see next what is meiosis.
Meiosis involves 2 successive divisions of a diploid 2N eukaryotic cell of a sexually reproducing organism that result in 4 haploid N progeny cells, each with half of the genetic material of the original cell. It is the nuclear division occurring in reproductive organs, resulting in gamete formation. Meiosis involves 2 successive nuclear divisions, Meiosis 1 and Meiosis 2. In this session, we shall learn about Meiosis 1. In Meiosis 1, there are 4 stages, namely prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1 is the longest and is further divided into leptotene, zygotene, pachytene, diplotene, and diakinesis. In the leptotene stage, individual chromosomes, each consisting of 2 sister chromatids, condense into visible strands within the nucleus. In zygotene, the chromosomes line up with each other into homologous chromosome pairs. At this stage, the synapsis pairing of homologous chromosomes takes place. The paired chromosomes are called bivalent or tetrad chromosomes. During the pachytene stage, the homologous chromosomes interchange equivalent sections of chromatids, a process known as crossing over or chiasma. This causes a crossing over with an exchange of genetic material between chromosomes. In the diplotene stage, the 2 homologous chromosomes separate from each other. In diakinesis, the paired chromosomes become short and thick and are distributed in the nucleus. In this stage, the nuclear membrane disappears. In the cytoplasm, the meiotic spindle forms between the 2 pairs of centrioles as they migrate to opposite poles of the cell. In metaphase 1, the spindles attach to the centromere of homologous chromosomes of each tetrad. As the process progresses into anaphase 1, the 2 chromosomes of each tetrad separate and start moving towards opposite poles of the cell as a result of the action of the spindle. In the final telophase 1, the homologous chromosome pairs reach the poles of the cell. A nuclear envelope forms around each chromosome set. Spindle disappears and cytokinesis follows. Cytokinesis divides the cytoplasm into 2 cells. After cytokinesis, each of the 2 daughter cells has a nucleus with a haploid set of replicated chromosomes. The 2 cells that are formed in meiosis 1 undergo similar stages in the next process and therefore called meiosis 2.
In meiosis 2, the amount of DNA, that is the chromosome number, is reduced back to normal by splitting the chromosomes. Meiosis II is also divided into 4 stages. They are prophase II, metaphase II, anaphase II, and telophase II. In prophase II, the centriole divides into 2 and each centriole migrates to the opposite pole. The nuclear membrane disappears and chromosomes with 2 chromatids become short and thick. During metaphase II, the chromosomes arrange on the equator of the spindle. The centromere divides into 2 and each chromosome produces 2 chromatids. The spindles are attached to the centromeres of the chromosomes. In anaphase II, the spindle fibers contract and chromatids move towards opposite poles. In telophase II, the chromatids completely migrate to the opposite poles and are now known as chromosomes. A nuclear envelope reforms around each set of chromosomes. Cytokinesis occurs, producing 4 daughter cells, each with a haploid set of chromosomes.